In this video today, I describe how viruses and transposons are both related to each other and perform functions which can be detrimental and in some cases advantageous to cells. Before we look at the fascinating example of the HIV virus, it's important to understand why viruses exist. Viruses cannot complete their life cycle without the assistance of cells. The virus is no more alive than a grain of sand. So viruses must infect cells in order to activate their metabolic machinery and generate progeny. All cellular life forms, ranging from bacteria through plants, fungi to animals, have their own sets of viruses which infect those cells. Why are viruses included in this chapter? As you'll see in the next few examples, viruses can adjust and alter the genomes of organisms simply by their life cycle. This figure here depicts the life cycle of a simple double-stranded DNA virus such as smallpox. The virus exists in two phases, one outside the cell and the other within the cell. The life cycle entails the entry of the DNA of the virus into the cell. That DNA then takes over the machinery of the cell for making proteins and copying DNA. Subsequently in the life cycle, the DNA and the proteins are combined to form the progeny viruses, which are then released from the cell, enabling the life cycle to continue. The processes which take place upon infection through to the release of the baby viruses, the progeny, are familiar to you. In this figure here, we have an alternative life cycle. This applies to the HIV virus, the one that causes AIDS. There are three important areas of note. First thing, the nucleic acid carried by the capsid is RNA and not DNA. That poses some challenges. As you know from previous chapters, DNA goes to RNA, which then goes on to protein. That is the central dogma of cell biology. Here, RNA has to be converted first into a hybrid molecule, and then that hybrid molecule has to be converted into DNA. The conversion of RNA into a hybrid molecule requires a special enzyme. That special enzyme, which is depicted here as a pink dot, is called reverse transcriptase. It's an enzyme which has the ability to copy RNA into DNA. Normally, cells don't have this enzyme. Therefore, the virus has evolved to carry a copy, at least one, in its capsid. The enzyme allows the reverse transcription of RNA into DNA, which is then converted to double-stranded DNA by the normal enzymes of a cell. The integrated viral DNA is activated early. In other cells, it may be activated many years later. The end result is the same. The DNA is converted to RNA through the process of transcription, and then the RNA is converted. Then some of the RNA is converted into proteins, including the proteins that make the capsid coat and copies of the very important reverse transcriptase. As the life cycle progresses, new capsids are made, which then leave the cell and repeat the life cycle. The important message here is that the genomes of target cells have been modified by the life cycles of viruses, especially retroviruses. Let's move on now to look at transposons, which through their behavior can also modify genomes. As we mentioned in the past, transposons can be classified into two categories depending on what happens to the DNA across which they move. If a transposon leaves behind a copy in the original location and it makes a new copy in the target location, We call that a copy and paste mechanism. So here you can see the red transposon has made a copy, and the copy has been inserted into the gold target DNA sequences. This leads to a massive increase in the number of transposons over time. The alternative mechanism is called cut and paste, where the original transposon removes itself from the DNA and then it inserts itself into the new DNA. There are some telltale signs left behind at the original location, but those are not important at this time. Transposons can fall into three main classes. The simplest are called simple transposons. They consist of a segment of DNA, the ends of which are mirror images of each other. Those mirror image regions are depicted here in red. Within the DNA segment, there's normally an active gene that encodes an enzyme called transposase. Once activated, the product of this gene produces an enzyme which has the ability to move the transposable element from one segment of DNA to another. An alternative arrangement that we've seen in other transposons is the compound transposable element. Very similar to the simple transposable element up here, the compound transposable elements, they contain more genes. So in addition to the transposase gene, which is necessary to move the transposon, these larger segments of DNA also carry additional genes. In this case, they carry a gene that conveys resistance to the antibiotic ampicillin. And in the third example at the bottom, we have a compound transposon which conveys resistance to the antibiotic tetracycline. So transposons, not only do they move themselves, they can also impart phenotypes on cells. So in this case, they make the cells antibiotic resistant should they be passed from one cell to another. Occasionally, the arrangement of the transposons is such that they flank a region of important DNA. When the transposons move, they take with them a segment of DNA and transpose that to a new location within maybe another gene, thereby shuffling the genome in the process. This can lead to rearrangements and production of novel proteins which did not exist before the transposition event. There is a third mechanism by which transposons move around the genome. That is called retrotransposition. Here we have double-stranded DNA in which the transposon has already inserted itself. This transposon will move by first copying itself into RNA and then converting the RNA into double-stranded DNA. That requires the enzyme reverse transcriptase. The double-stranded DNA can then be inserted into target DNA. This can lead to a rapid increase in the number of transposons that are positioned around the genome. Sometimes the insertion point can be within a gene, other times it can be outside a gene. This process is therefore comparable to how retroviruses move. Thus, there is some overlap between the mechanism by which retroviruses, HIV, and these retrotransposons accomplish their tasks. The concluding part of the chapter looks at how the human genome is arranged. In this slide, we're looking at one of the smallest human chromosomes and trying to decipher how the genes are arranged. Here in panel 4, we can see that a typical gene is mostly made up of introns with a few exons which are coded into protein. So most of the DNA within a gene is intronic sequence, which will be removed. In panel C, we can see that there are large regions of DNA interspersed between genes. This is intragenic DNA. so genes are quite sparse along the chromosomes. There are approximately 21,000 genes which code for proteins. A further 9,000 discovered so far that do not code for proteins but manufacture RNA. The largest gene is 2.4 million nucleotide pairs long, although the average size of a gene is 27,000 base pairs, and that would include the intronic sequences. If one maps out the DNA contained within a typical human nucleus, it is evident that only a small fraction is actually coding DNA. So about 2 to 3% of our DNA is coding for proteins. Another percent or so is coding for RNA. About 20% of our DNA is intronic, that is interdispersed between exons. So that accounts for about 22% of our DNA. The remaining DNA inside a human being is either repetitive DNA or unique DNA, which we label as intragenic, i.e. between genes. So this green segment is the DNA found between genes. And the remaining DNA appears to be repetitive DNA, which matches both transposable elements and retrotransposons. When comparing the genomes of organisms across the tree of life, we discover that there are great differences. When one compares DNA across different species, such as a yeast, which is a fungus, a fly, and an invertebrate, and a higher order animal, one discovers that there are many differences, reflecting different paths that the genomes of different organisms have taken across the eons of time. So in the case of yeast, the genes are very compact, there's very little intragenic DNA, and there's also little repetitive DNA. Compare that to the human, where we have lots of repetitive DNA, and we have lots of intronic DNA, and the red areas represent actual coding exons. One of the most revealing analysis of genomes came when the chimpanzee genome was compared to that of human. There's 98% similarity in the arrangement and positions of genes and repetitive sequences. So humans and chimpanzees differ by about 2% in terms of their DNA and only 1.2% with respect to their genes. So what makes a chimpanzee and a human so different? Well, as I told you last time, it's not just the presence of certain genes on DNA, it's also the timing at which these different genes are expressed. So in the case of a chimpanzee, the genes for making hair are activated, while those in humans are deactivated. Although the genes are present in both, the regulatory sequences have been modified to affect phenotype. So what makes you different to me? What makes you different to your neighbor? What makes you different to everybody else on the planet? Well, the answer seems to lie in not just differences between us in terms of alleles, blue eyes versus brown eyes, but also at the DNA level in differences in the base pairs. So if you compare one person from one part of the world, for instance an Aborigine, with somebody else, an Eskimo from another part of the world, they may have differences in their DNA outside genes. And those differences at single base pairs are the most frequent. And we call those SNPs, S-N-I-P-S. That's an acronym for single nucleotide polymorphisms. What is a single nucleotide polymorphism? Well, as you can see here, if you take 4 individuals on the planet and you sequence their DNA at the same regions on the same chromosomes, you will find subtle differences at certain base pairs. For instance, individual A has a G, whereas the other individuals all have an A at that position. Because there's a difference in at least one person, we label that as a SNP. In the second example in the middle here, we have a further difference between individual B and the other individuals. So that's labeled as a SNP2. And in position number 3, we once again have another difference. Regardless that it's in individual A, it is still a difference, and that's called a SNP3. So if we did this for the entire 3 billion base pairs of DNA that each individual carries, we should find a number of these SNPs. And those SNPs are key in making us different to each other, because some of them will naturally fall within the coding parts of genes or the regulatory parts of genes, therefore adjusting and coding for differences between us. In this final slide for this video lecture, we have an extreme case of exon shuffling. So here we have a piece of DNA, and from this piece of DNA in this particular tissue, we are manufacturing this messenger RNA. So you can see exon 1 isn't just a single segment of DNA that is included in the messenger RNA. It consists of repetitive sequences of DNA from which only one will be selected to be included in the messenger RNA. Likewise, from exon B, one of the green segments, just one, will be selected to be included in the messenger RNA. Same for exon C and exon D. Now, this is not a random process. What I mean by that is, at a particular time in the development of this organism and in a particular tissue, one of these will always be selected to be included in the messenger RNA. Now, what decides which one of these red segments will be included? That, we believe, is the combination of transcription factors and other regulatory proteins that will bind to the control elements for this gene. So the situation can be very, very complicated. So even though there are 38,000 possibilities for all these different cassettes, the actual number may be much less due to the fact that some of them are used in a few tissues at different periods of development. But this is an extreme case, once again, of exon shuffling and the potential that exists for one gene to code for many different types of messenger RNA. This concludes our first video lecture discussing Chapter 9, Part 2. Hopefully you are beginning to realize that we are what we are today as a result of past accidents or of past manipulations within our genomes. These have been caused by both transposable elements, by mistakes in mitosis and meiosis, and also by certain types of viruses.